A very good morning and warm welcome 10th standard for your online teaching learning program, Science P subject. Today we are going to start with lesson number 1, Heredity and Evolution. Before starting the lesson, I would like to tell you all, this lesson is a bit of scientific and technical terms, but quite interesting. It is about us, the genetic makeup, how the proteins are formed. Okay, there are 3 to 4 different types of mechanisms that we are going to study: transcription, translation, translocation, mutation. Okay, it's not new for you, but yes, something different we're going to listen for the first time, but few of its basics we have studied in previous standard. Okay, so I would request you all to pay attention, listen properly, and any doubts then we can clear in the next lectures. Okay, so kindly pay attention and let's start with this interesting lesson 1, Heredity and Evolution.
Students, when we hear this word heredity, immediately the first thing that comes to our mind is genetic material DNA and another genetic material, that is RNA. Now, students, you are very much familiar with this word as we have studied about hereditary in the previous class and also in detail about the DNA and types of RNA. We'll just look at the definition of heredity, that is transfer of biological characters from one generation to another. If you remember, students, we have also studied about one of the scientists who has contributed greatly in the field of genetics. He is Johann Gregor Mendel. In the year 1886, the principles given by him have also been studied by us, which are known as Mendel's principles of heredity. Hence, he was also known as the father of genetics. Along with Mendel, there were few other scientists who have also contributed in the field of genetics. We will just study about them. First one was Hugo de Vries. In the year 1901, he studied about mutation and the reasons behind mutation. Once again, we have studied about the mutation in the previous class, students, which is nothing but just the sudden changes that occur in the genetic material. Next, we have duo Walter and Sutton, who in the year 1902 proved that the genes are carried by chromosomes. Next, we have trio of Oswald Avery, Maclean McCarthy, and Colin McAloy. In the year 1944, they proved that except viruses, all the living organisms have DNA as the genetic material. Students, this science of heredity is very much useful for diagnosis, treatment and prevention of hereditary disorders that we have studied in the previous class. Also production of hybrid varieties of animals and plants, and in the industrial process where microorganisms are used. We will just look at the structure of DNA and types of RNA for revision, so that the contents that we are going to further study are clear to understand. Now, we all are aware that DNA or the genetic material is present in the cell of a body. Cell has a nucleus. The nucleus has something called as chromosome. This chromosome is made up of coiled DNA, and DNA molecules are made up of small segments called as genes, or whichever small segment or any segment of DNA is called as genes. We are familiar with this. Okay. Now, DNA being double-stranded, each strand is made up of small molecules which are known as nucleotides. This structure of nucleotide has three main components: molecule of a nitrogenous base of phosphoric acid joined to a molecule of sugar. The sugar present in DNA is deoxyribose. The four nitrogenous bases are adenine, guanine known as purine, and cytosine and thymine known as pyrimidine. Students, we have studied the function of DNA, transfer of characteristics from parents to offspring. This year, we are going to study another important function of DNA, that is synthesis of protein or protein synthesis. Material is RNA. There are three types of RNA: mRNA, messenger RNA; rRNA, ribosomal RNA; tRNA, transfer RNA. Messenger RNA. Messenger means who takes the messages from one place to another. So this messenger RNA takes away the messages from the DNA out of the nucleus. That is why it is called as messenger RNA. Ribosomal RNA, it is present inside the ribosomes. Transfer RNA transfers the amino acids to the different places. It transfers the things to the different places. That is why it is called as transfer RNA. Now, the next topic is central dogma. Now students, this is a new word for you. Central dogma means the synthesis of protein by DNA through the RNA. That is, the information for the synthesis of protein is present inside the DNA. It is to be transferred to RNA. It is to be passed on to the RNA. So a new RNA is formed inside the nucleus, and then this RNA takes the information from inside the nucleus in the cytoplasm. Because the first part of central dogma, that is transcription, takes place inside the nucleus, and the second part, that is translation, takes place inside the cytoplasm. So the proteins are synthesized by DNA through the RNA. This process is called as central dogma, and these proteins are synthesized as per the requirements of our body. So the first step is transcription. Now what is transcription? Transcription is the process of the synthesis of RNA from the DNA is called as transcription. Now how this process takes place? Now as you can see inside the nucleus, the DNA is present. The DNA is having the two strands. Now there is an enzyme called as RNA polymerase enzyme. Now because of this RNA polymerase enzyme, the two strands of DNA, they start getting separated. Now as they start getting separated, the messenger RNA is formed with the help of one strand of DNA. So only one strand of DNA is used in the process of transcription or in the synthesis of RNA from the DNA. Now there are free nitrogen bases present inside the nucleus. Now the messenger RNA, as we know, is a single strand structure. So it will be formed as per the complementary bases of any one strand of the DNA. Now suppose the lower strand is taking part in the formation of messenger RNA. So as you can see on the DNA strand, there are guanine, cytosine, adenine, thymine, cytosine, adenine. Now these bases we know that they form the pairs like adenine always bonds with thymine and guanine always bonds with cytosine. Now in RNA we know that thymine is replaced by uracil. Now we just look at the screen. Now the first base on the DNA strand is guanine. So the cytosine will come and form a bond with the guanine. Second one is cytosine. So the guanine will come and bond. Now third is adenine. Now adenine forms a bond with thymine, but as the RNA is being synthesized, so instead of thymine, it will be uracil which will bond with adenine. Next is thymine, again it will bond with the adenine. So this way the strand of RNA is formed with the help of one strand of DNA. Now this RNA has carried the information from the DNA and thus this is called as messenger RNA. It is going to carry the message from the DNA. Now this messenger RNA is formed inside the nucleus. Now the translation, the second step of protein synthesis takes place in the cytoplasm. Now this messenger RNA comes out of the nucleus and enters inside the cytoplasm. Now before this, students, I just want to give you the introduction of Dr. Hargobind Khurana. Now codon, as I said, they are the three bases. The triplet codon it is called as. So the three bases, when they come in the sequence, it forms a triplet codon. Now in total there are 20 codons. What is a triplet codon? It is a combination of three bases, and these bases they make different combinations. In total there are 20 such combinations or 20 triplet codons. Now one single triplet codon is for one amino acid. It gives the information for one amino acid. So when we have 20 triplet codons, that means we have 20 amino acids inside our body. Now this discovery of the 20 amino acids was done by an Indian origin scientist, Dr. Hargobind Khurana. He was the person who discovered that there are 20 amino acids in our body. So for this great work, he was awarded with the Nobel Prize in 1968 along with two other scientists. Now we will start with the translation. Now each messenger RNA is having thousands of triplet codons, that is the different combinations of three bases. Now as per the message on messenger RNA, the amino acids are supplied by the transfer RNA. Now how this transfer RNA will bond with the messenger RNA? Now the messenger RNA is having the triplet codons. Now these triplet codons will bond with the anticodons, that is the complementary bases, and these complementary bases are brought by the transfer RNA. It brings the basis for the formation of the codon and anticodon combination. That is why it is called as transfer RNA. So the transfer RNA has the anticodons. It comes near the messenger RNA and forms the base pairs. This is called as translation. Now these amino acids which are brought in by transfer RNA, they are bonded together with the codons present on the messenger RNA. Now during this process, there are ribosomes inside the cell, we know that very well, and inside the ribosomes there are ribosomal RNA, the third type of RNA. Now this ribosomal RNA, it comes and it moves from one end of messenger RNA to the other end of the messenger RNA. Now we had finished with the transcription and translation. Now the third, translocation, also goes along with translation. Now translation is what? Formation of codon and anticodon, right? That is the translation. Now when this ribosomal RNA brings in the amino acids, transfer RNA keeps on moving from one codon to another codon. Thus, this is called as translocation. It keeps on changing its place and thus it is called as translocation. So, these are the three processes of protein synthesis. Transcription takes place inside the nucleus. The message, the information from the DNA is copied on the messenger RNA. It comes out of the nucleus and enters inside the cytoplasm where the translation and translocation process takes place. This is the pictorial presentation of the translocation. As you can see, the transfer RNA is bringing the different amino acids near the messenger RNA, and the proteins which are formed, the amino acids, which are formed, they are forming a long chain, and this is done by the transfer RNA. The next phenomena we are going to study is mutation. What is mutation? And the disease caused by these mutations have been studied by you students in the previous class. We will quickly revise. We have studied that offsprings look alike their parents. Why? Because of the characters or the genes that is transmitted from one generation to another. Students, what happens is sometimes these genes are replaced or substituted, because of which the sequence gets disturbed and further the protein formed is different. These changes that occur in the genes is termed as mutation. Now these mutations could be minor, that is a single gene might be replaced, or even a complete codon might be replaced, which is known as a major mutation that you can see in the presentation on your screen. So basically mutations can be either minor or major depending upon the changes that occur in the sequence. Now we have studied the disorders like albinism, sickle cell anemia, hemophilia, night blindness, phenylketonuria, etc., which are caused by the mutations in an individual. So here we complete with transcription, translation, translocation and mutation.
So for today, this much is enough, and I hope concepts are clear to you all. Let's continue with the second part of the lesson next time. We shall start with evolution. Okay? Till then, thank you. Stay safe. Take care. See you soon.